Hey guys, welcome to Biotech View. Today we'll be learning about downstream processing, a complete outline of the process. The videos will be released in the form of a series covering all the important topics in downstream processing. So, downstream processing involves all the techniques and methodologies which are employed after a bioprocess is successfully completed, and it involves the product formulation, recovery, and much more. Let's have a look at the contents. We'll be covering all the important 5 stages of downstream processing, which is solid liquid separation, release of intracellular products, concentration, purification, and formulation. Now take a look at this pictorial outline depicting all the important processes involved in downstreaming process and observe keenly. The desired product is usually present in the culture broth. Now, this desired product can be an intracellular product or an extracellular product. So, if it is an intracellular product, a cell disruption mechanism must be employed in order to retrieve the product. But if the desired product is extracellular, we need to separate the solids and liquids which are present in the broth. And solid liquid separation can be performed by 4 mechanisms which we will be learning, after which concentration of the separated product is performed by certain mechanisms, and after that the purification of the product is usually performed by chromatography. We will be learning about the chromatographic techniques as well, and the final stage is the formulation stage. After formulation, we will receive the final product that is going to be released into the market. Let us now see, if the product is extracellular, so we need to separate the solids and liquids, right? So in order to perform that, we will be adopting 4 different kinds of techniques known as flotation, flocculation, filtration, and centrifugation. Let us now have a brief description of all the 4 processes. So, flotation is a mechanism in which a gas is introduced into the liquid broth, leading to the formation of bubbles. And now the cells and other solid particles will get adsorbed onto the surface of the gas bubbles. Now, since these gas bubbles are going to rise to the topmost foam layer, these bubbles can be collected and removed. Presence of certain substances called the collector substances will help in stable foam formation. So coming to the second one, it is flocculation, and flocculation is a mechanism in which the cells or the cell debris will form large aggregates which will settle down and that can be easily removed. The process of flocculation usually depends upon the nature of the cells and also the ionic constituents of the medium. Addition of flocculating agents like inorganic salts, organic polyelectrolytes, etc. is often employed in order to achieve appropriate flocculation. The third and the most commonly used technique for separating the biomass and the culture filtrate is filtration. The efficiency of filtration depends upon factors like the size of the organisms, presence of other organisms, that is contaminants, what is the viscosity of the medium, and the temperature of the process. Several filters like depth filters, absolute filters, rotary drum vacuum filters, and membrane filters are currently being used. Depth filter is composed of filamentous matrix like glass wool, asbestos, or filter paper. The particles which will be trapped within the matrix will not be able to move and the fluid passes out. Usually, filamentous fungi are separated using depth filters. The next kind of filters are absolute filters. Absolute filters contain specific pore sizes which are smaller than the particles to be separated. Now, bacteria from the culture medium can be easily removed with the help of these absolute filters. Rotary drum vacuum filters are the third kind of filters. These have been used for the separation of the broth containing 10 to 40% of solids by volume and also the particles which are in the size of 5 to 10 micrometers. These filters have been successfully used for the filtration of yeast cells and filamentous fungi. The equipment is very simple, it has low power consumption rate and is also very easy to operate. The filtration unit is consisting of a rotating drum that is partially immersed in a tank containing the broth. As the drum is rotating, it will pick up the biomass which will get deposited as a cake on the drum surface, and this filter cake can be easily removed. Fourth type are the membrane filters. Membrane filters contain specific pore-sized membranes which can be used for the separation of compounds. Clogging of the filters is a major limitation while using membrane filters, and these are of 2 types. Static filters and cross flow filters. In static filtration, the broth is directly pumped onto the surface and which will be filtered, whereas in cross flow filtration, the broth is pumped in a crosswise fashion across the membrane. This will reduce the clogging process and is hence better than the static filtration. The fourth technique employed for solid liquid separation is centrifugation. Centrifugation is based on the principle of density difference between the particles to be separated and the culture medium. Thus, centrifugation is mostly employed for separating solid particles from liquid phases, and this kind of separation is called as fluid particle separation. So, the centrifugation which is carried out at a laboratory scale is simple, but the one which is carried out at the industrial scale experiences major drawbacks and limitations. In order to overcome these limitations, continuous flow industrial centrifuges have been developed in which there is a continuous feeding of the slurry and the collection of the clarified fluid, while the solids which are deposited can be removed intermittently. Now the different types of centrifuges which are used currently are described briefly below. The first one is a tubular bowl centrifuge. This is a small and simple centrifuge and is the most commonly employed centrifuge at a pilot scale. Now tubular bowl centrifuge can be operated at very high centrifugal speed and can be run in both batch and continuous modes. The solids can be removed manually, that is, this method is not automated. Coming to the second kind of centrifuge is a disc centrifuge. Now disc centrifuge consists of several discs which will separate the bowl into different settling zones. The feed or slurry is fed through a central tube and the clarified fluid moves out upwards while the solids are going to settle at the lower surfaces. The third one is the multi-chamber centrifuge. This is basically a modification of the tubular bowl type and it consists of several chambers which are connected in such a way that the feed flows in a zigzag fashion. You can see the images which have been attached at the right corner of the slide and you will be able to understand the description in a better manner. Now since there is a zigzag fashion of arrangement, there will be different centrifugal forces in the different chambers. The outermost or the periphery chamber is going to have a centrifugal force which is very high. So all the smaller particles are going to settle down in the outermost chamber. The fourth one is scroll centrifuge or decanter. It consists of a rotating horizontal bowl which is tapered at one end. Now the decanter is generally used to concentrate fluids with high solid concentration. The solids are deposited on the wall of the bowl which can be scraped and removed from the narrow end. This brings us to the end of the first stage of downstream processing, which is solid liquid separation in case of an extracellular product. Thank you for watching the video and I hope this video series is going to help you all students out there. And I'm very eager to know how did you like the video. So please leave your suggestions in the comments section and stay tuned to Biotech View in order to get more videos in the series and also many more videos on biotechnology.